Voices from Oxford is talking today to an expert here in Oxford University, Professor Russell Foster, who is an expert on circadian rhythm. That's the rhythm that enables the daily cycle of sleep, awake, and so on to occur, and many, many other processes. And the reason we're doing that is that just recently the Nobel Prize in Physiology and Medicine has been awarded for the discovery of what are called clock genes. Now, Russell, you have to first explain to a general audience what exactly is a clock gene and what's happened.
Well, so we knew about some of these clock genes quite a way back, in about 1971.
It is quite a long story, isn't it?
It is a long story.
Yes.
Seymour Benzer discovered really the first gene as a result of mutagenizing flies, and then showed that these flies had abnormal sleep-wake rest activity profiles.
Right.
But the key sort of leap forward was, what are those genes actually doing?
Yes.
Now, what was so fascinating is that the first gene to be cloned, or the first gene to be identified, was called the PER, the period gene.
The period gene, yes. That determining the periodicity of the
oscillation. Yes,
indeed. Yes.
And at that time, though, into the, I suppose, late 80s, even up to the early 90s, people could not conceive that a multicellular organism such as Drosophila, the fruit fly, could have a subcellular molecular clock.
Right.
They thought that circadian rhythms, these 24-hour oscillations,
would be something to do with the body as a whole.
Well, they thought it was essentially cell-cell interactions, and in the same way that you can generate a swimming rhythm. We were talking to people who were interested in pure sort of network oscillators.
Yes,
and so the first thought about the PER protein was that it was a gap junctional protein, a protein that would unite and allow cells to connect.
Yes.
And it was only later on that it was realised that this couldn't be the case. And the breakthrough came from a lovely piece of work by an individual called Kathy Sawicki. Right. And what Kathy did was raise antibodies against the PER protein.
Okay.
And what she showed is that sometimes the protein was in the cytoplasm, and then other times it had gone into the nucleus.
So it can be part of a loop that goes all the way from the body, as it were, through into the nucleus to interact with the bit of DNA that is relevant.
That is exactly right.
Yes.
And
so you've got the beautiful feedback.
And so the concept of the feedback loop was born. A gene, the PER gene, produces a message that's translated into a protein. The protein is of course made in the cytoplasm, then enters the nucleus, and then turns off its own transcriptional drive.
Yes, indeed, that's right.
It was also appreciated pretty early on that PER could not act alone, although the first papers that came out were, the clock gene has been discovered.
Well, that was one of the questions I was going to ask you. How many clock genes are there, Russell?
Well, it depends on who you talk to. I mean, and of course, some of the genes have been duplicated. So in the fly, there's just sort of one form of PER.
Right.
But in mice, for example, there are 3 different forms of PER. Right. So we'll come to, I guess, mammals later on. But part of the Rosbash, Hall and Young team was to appreciate there's going to be more than just PER.
Yes.
And so independently, Young and Rosbash discovered PER's partner, Tim.
Okay.
And that then helped them understand that, in fact, the transcriptional drive on both of these genes was by yet more proteins. So this was clock and BMAL, or clock and cycle.
So you end up with really quite a complex network, which of course is, roughly speaking, what we've found for many other processes that interact with the genome, isn't it?
That's right. And so essentially the concept of the feedback loop was born, and the details over a decade were then refined. But I think the key point about the Nobel Award to these 3 individuals. And I feel it's absolutely justified, because these are the first individuals who genuinely discovered how clock genes would generate a feedback loop. And essentially the molecular clock.
Indeed, yes.
And from that, the mammalian work took place. Joe Takahashi.
That was in mice?
That was in mice, yes. And then, interestingly enough, fairly early on, different patterns of the human sleep-wake cycle.
Yes.
So familial advanced sleep phase syndrome. This is a condition where these poor individuals have to go to bed at around about 7:30 in the evening, and then are getting up about 4 o'clock in the morning.
Yes.
And that clock has gone much faster in those individuals.
Yes.
And it's a defect in one of the human PER genes. In fact, it's one tiny change.
Okay.
And it affects the stability of the protein and makes the clock go much faster.
But it goes faster.
Yeah.
So they end up, shifting slowly through the...
Well, yes, it's pretty tricky for them because they're sort of aligned in a fairly unstable way.
Yes.
But basically they're getting up early, very early, and going to bed very early.
So they're running in a jet lag problem all the time.
A little bit of one, yes.
Now it sort of brings me to the public interest in all of this. I mean, one might wonder why worry about circadian rhythm.
Yes.
Well, part of it is that when you or I or anybody else goes on a trip to East Asia or the other side of America, if we do get badly jet lagged, we're in trouble. Now, why does that happen?
Well, in us, we have a master clock within the brain ticking away. It rather like in Drosophila. And incidentally, I think it's extraordinary that the basic genes have been conserved across the entire animal lineage.
So these are very ancient.
These are very ancient.
Yes.
And you know, in species separated by 550 million years of evolution have this
and they're
extraordinary
all there.
conservation.
Sure.
So we have a master clock in the anterior hypothalamus in the brain called the Suprachiasmatic Nuclei.
No, long name, yes. I love the fact that you could pronounce it so easily.
SCN.
SCN, that's better, yes.
And what we thought was that this master clock would force 24-hour rhythms on the rest of the body. And then a chap called Uli Schibler from Switzerland discovered that actually lots of different cells, all peripheral cells, have clocks as well. So there's a massive hierarchy.
Exactly, yes.
And so part of the reason why jet lag is so ghastly is you're not just simply 5 hours shifted from London, let's say, to New York. It's that the whole circadian network is slightly out of sync.
So even your gut cells and your muscle cells, your skin cells are going to be all out of sync, and it's going to take time for all that to adjust.
Realign. And of course it's primarily exposure to the new light-dark cycle in the new time zone that forces the master clock to, let's say, shift to New York.
Indeed,
and then the clock is then sending out signals and
all over
which are trying to realign the rest of the...
So they're herding the rest of the cats into a suitable cycle. Well, look, thank you very much for talking to us about this, Russell. Just to finish, what are your feelings? I mean, it's lovely when one's own field is given a huge... Even if one doesn't get it oneself. It's lovely, and you deserve one, let me say that. I know your work. But it's lovely when one's field gets that sort of recognition, isn't it?
Absolutely splendid. And I think when I first entered this field, 25 years ago, there were about 60 people going to the meetings. And people have migrated in. I mean, Rosbash wasn't working on clocks. I know, and he came in and started working with Jeff Hall. And so I think it's just terrific. Now we go to meetings where there are 800, 900 people and more. And of course, clocks have been embedded across bioscience across the spectrum.
Indeed,
whether it's drug timing, cognition, psychiatry, and so many other fields.
So the implications are absolutely enormous.
Yes.
Thank you for talking to Voices from Oxford again. It's 4 years, I think, since we last spoke to you.
Yes.
And it's as exciting as ever.
It is.
Thank you very much, Russell.
Delighted, Dennis.